Assalamualaikum and a very good day. Today, our group will be presenting about synthetic carbohydrate.
My name is Amir Asfar bin Azrul, and for our presentation, I will explain about introduction and structure.
Assalamualaikum. My name is Muhammad Alif Hakimi, and for my part, I will discuss about application and health implication of 2-deoxy-D-glucose.
And I am Alia. I will be presenting about how to synthesize the synthetic carbohydrate and the properties of it.
So I will introducing about our compound, which is 2-deoxy-D-glucose, commonly referred to as 2DG. 2DG is a synthetic analog of glucose, meaning it shares a similar structure with glucose but has one key difference. The hydroxyl group at the second carbon in glucose is replaced with a hydrogen atom in 2DG. This slight structural change gives 2DG unique properties. It can mimic glucose and enter glycolytic pathways, the primary energy production process in cells. However, unlike glucose, it cannot be fully metabolized. This leads to inhibition of glycolysis, effectively disrupting energy production in cells. This characteristic make 2DG a powerful tool for scientific research and a promising therapeutic agent. It's currently being studied for its ability to target rapidly dividing cells, such as cancer cells and cells infected by virus. By cutting off their energy supply, 2DG has the potential to serve as a targeted treatment in this area. So, in the summary, 2DG is not just a metabolic inhibitor, but a compound with a significant implication for both research and medicine. So right now, I will explain about the structure of 2-deoxy-D-glucose or 2DG. 2DG comes from D-glucose, but the main difference is that it lacks a hydroxy group at the second carbon. Instead, this spot has a hydrogen atom, which gives 2DG its unique properties. In its stable form, 2DG has a 6-member ring called as pyranose ring, similar to the D-glucose. This ring is formed by an oxygen atom connecting carbon 1 and carbon 5. Now, for the functional groups, hydroxy groups are found on the carbon 3, 4 and 6. So, for the carbon 2 has a hydrogen atom instead of a hydroxy group. Carbon 1 contains an aldehyde group in the open chain form, but in the ring form, it becomes part of the structure. So, these small changes in the structure make a 2DG act differently in biological systems compared to regular glucose.
There are many ways to synthesize the 2-deoxy-D-glucose. Direct synthesis is mainly used as it is the most straightforward method and have a precise control to the selectivity. Through this method, the hydroxyl group at the carbon 2 position of the glucose can be reduced by replacing it with a hydrogen atom. Another method to synthesize this product is through indirect synthesis, in which the glucose is modified to an intermediate compound before the deoxygenation step takes place. Haloalkoxylation is another method where the hydroxyl group at the carbon 2 position can be substituted through nucleophilic substitution with a good leaving group such as bromine, which forms 2-bromo-D-glucose, followed by hydrogenation to remove the halogen. To give a more in-depth look on how the direct synthesis works, it begins with the protection hydroxyl groups by treating the D-glucose with a protecting agent, since the hydroxyl groups on the glucose is reactive and needs to be protected to prevent any unwanted side reactions. Next, to ensure the hydroxyl group at the carbon 2 is selected to leave the group, the protection group is reacted with a reagent like tosyl chloride or methyl chloride in the presence of a base to form a toslate or mesylate group at the carbon 2. Then, the toslate or mesylate group at the carbon 2 is replaced with a hydrogen atom through a nucleophilic substitution reaction by reacting with a strong reducing agent like lithium aluminium hydrate. Afterwards, the protecting groups need to be removed by treating the intermediate with a deprotection agent like dilute acid. Lastly, the crude product is purified through recrystallization or chromatography to isolate high purity 2-deoxy-D-glucose. On the screen, we can see the chemical equation on how the direct synthesis takes place. For the physical properties of the 2-deoxy-D-glucose, the appearance is a white crystalline solid or powder. The molecular weight is 164.16 grams per mole, and it is soluble in water and in polar organic solvents like ethanol. The melting point is around 147 to 150 degrees Celsius, and it is optically active due to its chiral nature, and it rotates plane polarized light to the right, similar to glucose. For the chemical properties of the 2-deoxy-D-glucose, the structure and functional group is similar to glucose because of the hydroxyl group at the carbon 3, carbon 4, and carbon 6 position, and these functional groups contribute to its reactivity. However, it lacks the hydroxyl group at the carbon 2 position. The 2-deoxy-D-glucose can be reduced to produce sugar when treated with reducing agents. It can also be oxidized, but the lack of hydroxyl group at the carbon 2 makes it less reactive in some oxidation reaction that the glucose might undergo, such as forming glucuronic acid. The 2-deoxy-D-glucose also has a weak acidic properties despite it is not strongly acidic or basic in nature. It is also relatively stable under normal conditions, but it may undergo degradation or rearrangement under extreme pH conditions or high temperature.
Okay, so for the application of 2-deoxy-glucose, the structural modification impair its metabolism, allowing 2-DG to interfere with various biochemical pathways that utilize glucose. Due to its unique properties, 2-DG has a range of application across multiple fields, including medicine, research, and also diagnostic. For the first application, pharmaceutical and biotechnological industry, cancer therapy, used as an adjunct treatment to target glycolysis, depend cancer cell, and changing the effect of chemotherapy and radiotherapy. For the second application, medical imaging and diagnostic research used to study glucose uptake and metabolism in tissue, aiding in the understanding of the metabolic disease. For the third application, industrial biotechnology and food. For the fermentation process, a tool for controlling glucose utilization in industrial fermentation, which can improve yield and reduce costs. Her application of 2-deoxy-D-glucose has significant potential as a therapeutic agent and research tool, but its effects on health involve both beneficial application and associated risks. Below is a detailed analysis of its health application. For the cancer therapy, can enhance the efficacy of chemotherapy and radiotherapy, and then offer the target approach for tumor reliant on aerobic glycolysis. For the neurological disorders, study for its potential to modulate glucose metabolism in the brain, and then may be beneficial in conditions involving neuronal hyperactive or energy dysregulation, such as epilepsy or Alzheimer's disease. That's all.
That is all from us. Thank you.